点晴朗，内心的回响，守候的遗忘，都在那里收藏。灿烂的阳光，播种力量，明亮的月光，温暖梦想，梦放牧在心上，心辽阔无疆，爱打开了翅膀，爱纵情飞翔。心上，有爱的地方就是生命的天堂，天堂。灿烂的阳光，播种力量，明亮的月光，温暖梦想，梦放牧在心上，心辽阔无疆，爱打开了翅膀，爱纵情飞翔。
It is now October 25th, 2020 in the Christian era. The development of a vaccine against the pathogen of the new coronavirus infection will be described. It's important, so please listen carefully. The pathogen is a coronavirus, and we are now developing vaccines around the world. But the truth is, I realized that no one in the world knew what they were developing. Therefore, this time, as part of the special Bioscience Lecture, I decided to briefly explain what vaccines the United States and the United Kingdom are developing so that even elementary school idiots can understand. There is a phenomenon called the RNA polymerase calibration mechanism of coronavirus. The new coronavirus is an RNA virus. The RNA virus is constantly mutated because it also contains an exonuclease, an enzyme that has calibration function that corrects errors that occur during replication of viral RNA. It changes the genomic information without any problem. However, this theory does not apply as the pathogen of the new coronavirus infection COVID-19 is coronavirus SARS-CoV-2. It frequently causes mutations in the body after infection. Unlike other coronaviruses, there is an abnormality in the RNA polymerase calibration mechanism. The reason is a mystery and unknown. Mutation rates in nature have not been investigated as closely as HIV, and with some exceptions the reality remains unclear. However, RNA viruses are generally considered to be as highly mutagenic as HIV. First, with the exception of some retroviruses, the mutation frequency during replication of viruses in cells is equivalent to that of HIV. And the mutation potential during replication is thousands of times higher than that of microorganisms that use DNA in the genome. Since RNA virus proliferates actively in the host, it has a large number of replication cycles per year. For this reason, RNA viruses are considered to be the fastest changing organisms in nature. Is wrong. Coronavirus is exceptionally unlikely to mutate among RNA viruses. Professor Miyazawa, Institute for Frontier Medical Sciences, Takayuki Miyazawa. Virus, Kyoto University. The causative virus of AIDS is officially the human immunodeficiency virus. Considering the infection efficiency of this virus when a male HIV-infected person has sexual intercourse without a condom, the infection is established by sexual intercourse with an HIV-negative woman approximately once every 100 to 1,000 times. The infectivity of HIV is that much. If there are 100 virus particles, it does not mean that up to 100 cells will be infected. There is also interference with retroviruses such as HIV, a resistance factor to intracellular HIV, which is believed to be infectious in 1 in 100. Normally, infection should not be established unless a certain number of viruses invade the cells in a population. But in the case of this new type of coronavirus infection, there are many strange phenomena. There are many reports that people are infected when they are talking at a distance of several tens of meters due to close contact, or they are infected just by having a conversation nearby. For example, in Middle East respiratory syndrome MERS, coronavirus infects only 1 in 100,000 virus particles. In other words, although the gene structure is similar to that of coronavirus SARS and MERS, it is better to position the COVID-19 pathogen as a completely different type of pathogen. However, in theory, emergency measures are taken based on the basic idea that if the viral load is reduced to 1/100th of the peak, the virus will not be infected. In other words, the Centers for Disease Control and Prevention in major developed countries should take immediate measures to reduce the number of infected people to about 100 million, even if all 8 billion humans are infected. Become a virus that proliferates using proteins in human cells will have some toxicity if the drug that inhibits functional proteins in human cells is used as a therapeutic agent. Especially when used for a long period of time, it causes fatal damage. Although it is an RNA polymerase inhibitor developed in Japan for the treatment of Antigen and the new strain of influenza, animal experiments have shown that when used in pregnant women, the probability of developing malformed children is extremely high. It is thought that this is because it adversely affects the process of transcription when making proteins from the DNA of the human body. It is thought that there is a similar view on the nucleic acid analog called remdesivir, an American drug for the treatment of Ebola. Therefore, it is necessary to find a promising therapeutic agent that inhibits the specifically expressed protein required for the virus itself to grow. Since this can be discovered accidentally rather than predicted from past experience, it is possible that nothing will be found or something will be found in the future. However, at present, The growth of the virus is extremely high, and the pathogenic malignancy and mortality rate are also high. So there is no time to select a therapeutic agent from the beginning and verify the synthetic method and therapeutic effect. Therefore, in order to avoid the danger of human extinction, it is first necessary to find a way to stop the growth of the virus. The distribution of European stocks grows at an alarming rate. From London, regarding mutations in the new coronavirus on April 29. The Los Alamos National Laboratory in the United States identified 14 mutations in spike proteins, and one of them, G614G, a so-called European strain, started in early February. He published a peer-reviewed paper pointing out that the infection spread in Europe and spread all over the world. The paper states the distribution of D614G is increasing at an alarming rate, demonstrating the fitness advantage of being able to spread more quickly than the original Wuhan strain. Since spike proteins are important targets for vaccines, fast mutations also have a significant impact on vaccine development. Patients infected with European strains seem to have a high viral load, but in terms of hospitalization rate, it seems that there was no big difference between Wuhan strains and European strains. However, even if you infect the Wuhan strain and produce antibodies, you may infect the European strain. The graph below shows the trends of Wuhan strains orange and European strains blue, which have become the mainstream of the epidemic. When the virus propagates, it copies the genetic information. But at this time, the character spaces may be copied incorrectly. This is a mutation. The new coronavirus has a proofreading function, viral proofreader, NSP14, that finds these mistakes and cuts out the characters to replace them with the correct characters. No one knows what the future holds. It's an unpredictable problem. But, as far as I read the research papers published so far, for the amateur author, the new coronavirus repeats base mutation at a pace of once every 2 weeks depending on the environment such as racial immunity, climate, public health intervention, etc. It is considered that only those that adapt to the environment die due to the selection, that is, the competition for survival, and those that adapt to the environment remain. The viral condition is already survival of the fittest, and in biological evolution terms, there may be a phenomenon in which the most environmentally friendly one is guaranteed a chance to survive in the competition for survival. In other words, if there are too many mutations in the gene, there is a high probability that the virus itself will not be able to multiply. It is possible that the virus has sprung up and infects all humans and other animals at an alarming rate, and the increase in mutant species has diminished for about half a year now. However, it cannot be denied that there is a possibility that a pathogen with an instant death level will suddenly appear in all human beings, a high degree of malignancy that is completely different from the current one. Because when infected with this current pathogen, there is a mechanism that mysteriously prevents human immune function from functioning normally. How does the body distinguish between autologous mRNA and foreign mRNA? In fact, it is highly possible that the answer is there is no such mechanism. Clinical trials of mRNA vaccines have already begun. One that challenges in medical application of mRNA are that mRNA is extremely unstable in vivo, especially extracellularly, and that it has strong immunogenicity through the innate immune mechanism. In the 2000s, through basic research on modified nucleic acids such as pseudouridine, thiolated uridine, methylated uridine, and cytidine, it has become clear that proper nucleic acid modification of mRNA can control immunogenicity in vivo. Various modified protocols have been reported. Living body does not recognize foreign mRNA as a substance, and the intracellular localization of mRNA is that in order to use mRNA as a drug, it is sufficient to deliver the mRNA in a stable form to the appropriate place in the cell, and then the protein will be expressed through the original translation mechanism. It seems to be point. When a block copolymer is mixed with a nucleic acid molecule such as mRNA, the nucleic acid molecule is encapsulated in a condensed state, and a nanoscale aggregate micellar carrier whose circumference is covered with PEG is formed. Although it is a problem common to DNA and other nucleic acid drugs, in order for mRNA to become a drug, it is necessary for mRNA to be taken up by individual cells and express its function. Therefore, it is necessary not only to deliver mRNA to the vicinity of the cell and allow it to be taken up by the cell, but also to release mRNA rapidly in the cytoplasm, which leads to protein expression. After the PEG carrier is taken up by the cell by endocytosis, rapid intracytoplasmic translocation of the carrier occurs in response to the endosomal acidic environment in nervous system tissue, which is one of the important targets of mRNA. Administration of mRNA-loaded micelle-type carrier PEG-P ASP-DET mRNA into the subarachnoid space of the spinal cord stabilizes the carrier in the cerebrospinal fluid. By spreading, protein expression was obtained in a wide range from the brain to the lumbar spinal cord. Naturally, mRNA has a wide range of indications other than the nervous system, and has the common property of being widely introduced into cells of target tissues and organs. When this property is analyzed in detail by hydrodynamics administration to the liver, protein expression in individual cells tends to be lower than that in pDNA administration, but expression is observed in nearly 100% of cells in the target organ. Matsui et al. Messenger RNA-based therapeutics for the treatment of apoptosis-associated diseases. Scientific Reports 5-15810, 2015, as a means for introducing a transcription factor into a cell. It is impossible to introduce the transcription factor as it is, and it is necessary to take the form of gene transfer. Of course, virus vectors can also be introduced. The single-strand RNA possessed by the new coronavirus, SARS-CoV-2, etc., takes the structure of double-strand RNA in the process of replication after invading the host cell. After that, immunity is induced by activating antigen-presenting cells that recognize double-stranded RNA. Immune cells detect the presence of double-stranded RNA in the body as abnormal. By taking advantage of this mechanism, it is possible to develop an mRNA vaccine that strongly induces immunity. Overseas, the development of mRNA-based vaccines for COVID-19 has taken the lead, and many of them use lipid nanoparticles (LNP) as the drug delivery system (DDS). The problem is that even if the vaccine is administered to the bloodstream, it is presumed that the expression level of the protein as an antigen is low in the cells of the airway epithelial mucosa, which generally have low blood flow. Furthermore, even if a peptide that is a protein fragment of the cell binding domain of the virus spike protein is used as an antigen, the actual preventive effect of the virus is structurally impossible due to the large number of pathogen mutations in the binding to the antibody. It is conceivable that, moreover, no pathogen that spreads worldwide in a short period of time has ever been seen in human history. This means that the infectivity and proliferation of the pathogens of the new coronavirus infection are extremely high compared to HIV and influenza, although the detailed mechanism is unknown. Mutations are an essential phenomenon for a virus to survive. This is because in order to survive, it is necessary to change the amino acid sequence so that it can adapt to the environment. However, not only the amino acid sequence but also the spike protein is glycosylated by being modified by a plurality of carbohydrates. This plays an important role in the binding of the three-dimensional structure of the protein to the antibody. Human beings are trying to find out the characteristics of the virus only in the amino acid sequence of the virus by genetic analysis, but they have noticed that it is a big mistake. However, the detailed mechanism of the modification reaction of glycosylation is unknown. Regarding the change in amino acid sequence, It is not clear at all what kind of functional change each point mutation causes in the corresponding structural protein or non-structural protein. I don't know if the mutation changes the function of the viral protein. Polymer nanotechnology for nucleic acid delivery. Nucleic acid drugs have a strong therapeutic potential for treatment of many intractable diseases, including cancer. However, their bioavailability seems to be miserable owing to their high susceptibility to enzyme degradation and negatively charged macromolecular structures that hamper their cellular entry. To overcome these bottlenecks, a variety of delivery vehicles, such as ligand-nucleotide conjugates, lipid nanoparticles, and polymer nanoparticles, have been developed for successful nucleotide therapeutics. This article describes the design strategies of synthetic block-copolymer-based nucleic acid delivery systems, particularly highlighting smart polyion complex pigmy cells that can exert the desired functions in response to biological microenvironment signals. Also, our recent results related to small interfering RNA, siRNA, and messenger RNA delivery are introduced with details. Notably, the ligand-installed pigmy cell has achieved efficient tumor-targeted siRNA delivery through systemic route, leading to the significant anti-tumor activity in subcutaneous tumor models. I explain about coronaviruses. Coronaviruses contain positive stranded RNA with ca. 30 kilobits as a genome, which is wrapped by the envelope, and constitutes nidovirals together with arteriovirity. The feature of viruses in nidovirals is the unique structure of the mRNA set, called 3-coterminal nested set. Coronaviruses have several to more than 10 different species of subgenomic mRNA, and generally only the OFR located in the 5' end of each mRNA is translated. The 520 kilobits of the coronavirus genome or mRNA1 consists of two ORFs, 1A and 1B. Between that there is a unique RNA structure called pseudoknot. From mRNA1, 1A as well as 1A plus 1B are translated. The latter 1A plus 1B results from the translation due from those two proteins, totally to ribosomal frameshifting facilitated by the pseudoknot structure. 16 proteins are produced as a result of autocleavage by the proteases included in 1A protein. Those proteins exhibit different functions, such as RNA-dependent RNA polymerase, helicase, proteases, and proteins that regulated cellular functions. mRNAs smaller than mRNA2 translate in general the structural proteins, nucleocapsid, and protein, spike S protein, integrated membrane M protein, and envelope E proteins. Those proteins assemble to the vesicles located from Golgi intermediate compartment, and virions bud into the vesicles. Those virions are released from infected cells via exocytosis. Viruses are found in all flora and fauna, protozoans, fungi, bacteria, rickettsia, chlamydia, and mycoplasma. That is, the virus host is distributed in all species except the virus. Most coronaviruses are livestock and pets, including humans. It mainly causes digestive and respiratory illnesses, but the pathogenicity mechanism is not well understood. Coronavirus (CoV) is a virus belonging to the order nidovirales along with arteriovirus, and its genome is an RNA virus with an envelope of about 30 kilobits plus strand. The characteristic of CoV is the structure of mRNA, which is composed of several mRNAs that extend from the 3' side to the 5' side of the genomic RNA with different lengths in each mRNA. It has a leader sequence at the 5' end of genomic RNA. From its structure, it is speculated that mRNA is formed by discontinuous RNA synthesis, but there are still two hypotheses about the mechanism. There is no definitive experimental proof of which is correct. Translation of viral proteins is generally translated only from the ORF located at the 5' end of each mRNA. The 5' of about 20 kilobits of genomic RNA mRNA1 consists of two ORFs, 1A and 1B encode 802 kDa. Between these ORFs, there is a region with a complex cubic structure called pseudo-PN. So there are cases where translation is terminated by only 1A protein and cases where 1A plus 1B fusion protein is synthesized by PN. The 1A plus 1B protein is cleaved into 16 regulatory proteins in addition to acting as AROTEs, and RNA polymerase proteins that suppress cell protein synthesis have also been identified. Basically, structural proteins are translated from mRNA to and below. Coronavirus (CoV) is only known as one of the causative viruses of human nasal colds. And research in the medical field has been limited because there was no virus that causes serious illness in humans. There are few past research papers. First, the genome of coronavirus and the structure of mRNA will be described. The viral genome consists of approximately 30 kilobits, and each mRNA is several mRNAs that extend from the 3' end of the genomic RNA to the 5' end with different lengths. Consists of. This structure is called a 3' coterminal nested set, and is a feature shared not only by CoV but also by viruses of the order nidovirus. Next, the mechanism of mRNA synthesis of coronavirus will be described as one pattern. A strand RNA complementary to it is synthesized from the genomic RNA, and a free leader RNA complementary to the 3' end of the strand RNA is synthesized. A TRS consisting of the number base of the 3' end region of free leader RNA binds to a site complementary to the TRS on that strand RNA. And the elongation of subgenomic mRNA (sgmRNA) continues to the 3' end. Complementary strand RNA is synthesized from sgmRNA. As another pattern, complementary strand RNA synthesis for each mRNA is performed from genomic RNA. When it reaches the TRS site during synthesis, it binds to the TRS of the leader RNA. RNA synthesis continues, and a strand RNA complementary to mRNA is completed. mRNA synthesis is performed from each mRNA complementary RNA for both A and B. The right-pointing and left-pointing arrows indicate the RNA synthesis direction. Translation from genomic RNA mRNA1 shows that genomic RNA has giant ORF 1A, 1B at the 5' end, with pseudo-PN in between. Protein translation usually ends at the 1A stop codon, but with a 20 to 30% chance, a frame shift occurs due to the PN structure. The 1A stop codon is not read, and the translation continues to 1B, 1A plus 1B. The protein is ready. These giant proteins are cleaved into 16 types of proteins by their own proteases. Since RNA polymerase does not have the calibration function that DNA polymerase has, it is difficult to restore it when a mutation occurs. Therefore, RNA viruses have a higher mutation frequency than DNA viruses. Vaccinations are broadly divided into live vaccines and inactivated vaccines. For live vaccines, select a pathogen with weak pathogenicity and continue culturing for many generations, hundreds of generations depending on the pathogen, to produce symptoms. The pathogenicity is weakened or eliminated to the extent that occur. Next, I explain vaccine production method. Pathogens may be attenuated by passage in the following heterologous hosts. Tissue culture. Growing chicken eggs. Surviving animals introduce a group of primary cultured microorganisms into a heterologous host. Some of the microbial community are likely to produce mutations that allow infection of heterologous hosts. Over time, it acquires many mutations to grow in the host, making it significantly different from the primary group. This facilitates the elimination of pathogens and the formation of immune memory cells by the immune system of the source host, increasing When it is reintroduced into the original host, it does not grow as well, i.e., mutant D, as in a heterologous host. The ability to protect patients against infection with highly pathogenic similar species of microorganisms. Administration. Live vaccines use live viral particles or bacteria with extremely low pathogenicity. Replication is very slow, but does not require much booster as it replicates continuously and produces more antibody than at initial vaccination. Vaccines are produced by tissue culture to select less pathogenic strains. Mutations. Administration to immunocompromised individuals may be contraindicated. There is a small risk of reversal of pathogenicity, but the risk is small for vaccines produced by targeted removal. A method for producing a vaccine will be described. Vaccine production requires a large increase in non-pathogenic pathogens, and currently there are 4 methods: hatched egg culture method, animal inoculation method, cell culture method, and gene recombination method. 1. Hatched chicken egg culture method. It is a manufacturing method mainly used for influenza vaccines. This method is used in hatched chicken eggs, fertilized eggs. A small amount of influenza virus is inoculated into the allantois cavity to propagate the virus, and serous fluid, serous urine, liquid, is collected. 2. Animal inoculation method. 3. Cell culture method. 4. Genetic modification method. The virus gene was inserted into a special cell that had been propagated in advance, and only the protein involved in the antigenicity of the virus was made into the cell, and then these were taken out and purified. Fig. 4. The advantages are that the production period can be significantly shortened and that vaccines can be produced safely because no infectious virus is used in the production. This technology has been used in the production of hepatitis B vaccine using yeast cells and has already been put into practical use. Polymer nanotechnology for nucleic acid delivery. Nucleic acid drugs have a strong therapeutic potential for treatment of many intractable diseases, including cancer. However, their bioavailability seems to be miserable owing to their high susceptibility to enzyme degradation and negatively charged macromolecular structures that hamper their cellular entry. To overcome these bottlenecks, a variety of delivery vehicles, such as ligand nucleotide conjugates, lipid nanoparticles, and polymer nanoparticles, have been developed for successful nucleotide therapeutics. This article describes the design strategies of synthetic block-copolymer-based nucleic acid delivery systems, particularly highlighting smart polyion complex pigmy cells that can exert the desired functions in response to biological microenvironment signals. Also, our recent results related to small interfering RNA, siRNA, and messenger RNA delivery are introduced with details. Notably, the ligand-installed pigmy cell has achieved efficient tumor-targeted siRNA delivery through systemic route, leading to the significant anti-tumor activity in subcutaneous tumor models.